Hello friends, in the last class we have discussed about NEET Zoology, chapter-wise conceptual analysis and approximate weightage. In today's lecture, we will be discussing about NEET Botany, chapter-wise conceptual analysis and approximate weightage. At first, we will discuss about class 1 or class 11 NEET Botany. NEET Botany class 1. No. Broadly, it consists of 4 units. Number 1, diversity in the living world. In the living world. Number 2, structural organization in plants. Number 3, cell structure and function. Number 4, plant physiology. There are the 4 units under class 1 or class 11 NEET Botany. Diversity in the living world, but that is based on expert analysis and over the years prediction. Diversity in the living world may occupy around 8%. Whereas structural organization in plants may occupy around 9%. Whereas cell structure and function may occupy around 15%. Respect to plant physiology, which is important, it is around 17%. Okay. So it is very clear that the cell structure and function, particularly the plant physiology, are very important in NEET examination point of view. I have already discussed with you that this prediction will give us enough confidence and direction that which chapters that we must focus, and we have to regularly revise and take mock test, so that accordingly we can get maximum marks. Okay. And first, diversity in the living world. Okay. This diversity in the living world again have different sub chapters. Diversity in the living world. It deals about basic features. For example, what are the characteristics of living organisms? What makes a life? For example, there are certain characters which are essential for a living organism. Say growth. Say for a living organism, the essential character is growth, reproduction, metabolism, consciousness. For example, characteristic features. Characteristics of life. In the sense, say growth, reproduction, metabolism, consciousness. These are the fundamental characters for our life. So we have to discuss at least in brief about growth, reproduction, metabolism and consciousness, which are the essential characteristics of life. Then coming to the other topics, actually this diversity in the living world broadly deals with the basics. Say the basic aspects of biodiversity, for example. Then the taxonomical categories. It's like definitions. We have to know certain basic words. For example, taxonomic category, say in a hierarchy, for example, kingdom, phylum, class, order, family, genus, species. We have to know at the basic level about taxonomical categories. Say nomenclature. What exactly the nomenclature is, what identification, classification, etc. It's all about basics of taxonomical issues. Okay. Apart from this, we have to know about taxonomical aids. Taxonomical aids. Say like herbarium, zoological parks, botanical gardens. This unit deals with about not like definition level. The basic understanding is enough at the moment. Say the hierarchy, particularly with respect to taxonomy categories, and what are the living characteristics. This is very important. I have mentioned, okay, and remaining just at the basic definition level, but conceptual understanding, okay. Then other contents in the diversity in the living world deals with biological classification, biological classification. This biological classification means, say, Aristotle is considered as the first person to classify organisms on scientific basis. Then in Linnaeus times there is a two kingdom classification like plant and animalia. At present we will be following about Whittaker classification, which is a 5 kingdom classification. He has divided the living organisms into 5 kingdoms like kingdom Monera, Protista, Fungi, Plantae and Animalia, based on cell structure, body organization, mode of nutrition, reproduction and phylogeny. So we have to discuss at length about certain basic issues for better understanding. Biological classification like Whittaker deals about 5 kingdom classification. Say Monera, kingdom Monera, Protista, Fungi, Plantae and Animalia. 5 kingdom classification, okay, by Whittaker. In examination point of view, they may ask you what are the parameters, what are the basis that Whittaker has included for this classification. As I mentioned, cell structure, body organization, mode of feeding, reproduction, phylogeny, for example, phylogeny relationship. Based on these characters, he has classified the entire organisms into 5 kingdoms: Monera, Protista, Fungi, Plantae and Animalia. Say, in brief, prokaryotes, all prokaryotes comes under Monera. All prokaryotes comes under Monera. Single cellular, unicellular eukaryotes comes under Protista. Unicellular eukaryotes. So conveniently we can say, starting from Protista and up to Animalia, they are all eukaryotes. They are all eukaryotes. The thing is, difference based on the complexity of organization. And I told you already, Protista is a unicellular eukaryote. Fungi, then again multicellular or loosely arranged tissues. Plantae animal, like that means tissue, organ level, organ system level. Likewise, starting from Protista and up to Animalia, they are all eukaryotes, and all prokaryotes come included under Monera. Let us have a look at least in brief about different kingdoms. Okay, kingdom Monera. Broadly it includes, as I mentioned, prokaryotes. Say for example, archaebacteria it includes. Archaebacteria and eubacteria like cyanobacteria. See, kingdom Monera, we have to discuss about, we have to know that what are the representatives of Monera. Say the archaebacteria, eubacteria like cyanobacteria. Just at least we have to know which organisms or which group of organisms comes under which kingdom. This is very important, at least in basic sense we have to know. Okay, then kingdom Protista. I told already they are unicellular eukaryotes. The significance of the kingdom you have to know. Unicellular eukaryotes. It includes like organisms like dinoflagellates, chrysophytes, euglenoids, protozoans. Okay. Protozoans are different, like amoeboids, ciliated, flagellated, like this. Flagellated, sporozoans. Sporozoans is malarial parasites, like that. Slime molds. These are all different groups. Comes under kingdom Protista. Dinoflagellates, chrysophytes, euglenoids, protozoans, slime molds. Okay. Then coming to the fungi. Kingdom fungi, even to at least add, as I mentioned, we have to know at basic level in brief. Kingdom fungi. The significant feature of kingdom fungi is it had a cellulose with chitin. That means a cell wall with chitin is very important. Okay, we will discuss about in later. That means while we discuss about the third unit, that means cell. In kingdom fungi, it includes different classes like phycomycetes, ascomycetes, basidiomycetes, deuteromycetes. Okay, this kind of fungi have different classes like phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. Okay, next coming back to the kingdom plantae. Kingdom plantae. Broadly, it has 5 subcategories. Algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae, there are different classes. In last class, we have discussed about kingdom animal. Okay, in case of with respect to kingdom plantae, like algae, it have different classes like chlorophyceae, phaeophyceae, rhodophyceae. Bryophytes includes liverworts and mosses. Pteridophytes include horsetails and ferns. Gymnosperms are here the ovules are naked. The significant characters, ovules are naked. Plus angiosperms is what we call flowering plants. Flowering plants. Here, unlike gymnosperms, it have pollen grains and ovules enclosed in specialized structures called flowers, and seeds are present in the fruit. Pollen grains and ovules, they are present in a flower. Bentham and Hooker classification is very important for angiosperms. And these angiosperm again have 2 classes: dicotyledons, monocotyledons. Dicotyledons, monocotyledons means single seed with 2 cotyledons. 2 seed with. Here monocotyledon seed with 1 cotyledon. It's about the broad kingdom, that means classification of kingdom plantae. And algae have different classes like chlorophyceae, phaeophyceae, rhodophyceae, whereas bryophyceae includes liverworts and mosses. I am giving peripheral analysis and conceptual wise brief account for understanding which topics and what to stress and regarding the direction. Okay, pteridophytes include horsetails and ferns. Gymnosperms and angiosperms include only 2 classes: dicotyledons and monocotyledons. Okay, then it's about kingdom plantae. Next, it's all about diversity in the living bold. Okay. It accounts nearly 8% as I mentioned. Okay. The second unit is structural organization in plants. Structural organization in plants. Broadly, it includes two sub chapters, like morphology of flowering plants, anatomy of flowering plants, anatomy of flowering plants. Morphology of flowering plants. The name it implied. We'll be discussing about morphology of roots, stem, like root modifications, for example, tuberous roots, for example. Stems, different stem modification like aerial stem modification, subaerial stem modifications, for instance, underground stem modifications. That is a basic level, okay? Leaves, like what is phyllotaxy, venation, like that, okay? Then inflorescence, flower, and apart from this, we have to discuss about certain, three important families, semi-technical aspects of a flowering plant, and also some important families like Fabaceae, Solanaceae, Liliaceae. These all comes under morphology of flowering plants. As I mentioned, the general description of a plant we have to understand. What are the important parts? What we call technical description of important parts. We have to understand. What is the calyx? What are sepals? What are petals? Likewise, at least at the basic level. Whereas anatomy of flowering plant, it deals with anatomy. For example, tissues. Say tissues, like for example, there are two important tissues like meristematic tissues. We'll discuss. We will take a separate chart. Okay. In anatomy of flowering plants, discuss about tissues. Tissues are again here two kinds broadly. Meristematic tissues like apical meristems, lateral meristems. Okay. Then permanent tissues. Here again of two kinds, simple and complex. Simple, like parenchyma, collenchyma, and sclerenchyma. Complex includes xylem, phloem. So, to understand the basic description, what we call features of meristematic, what exactly meristematic tissues, the permanent tissues. Its categorical, that means classification is very important. In examination point of view, they might ask you, sclerenchyma is what kind of tissue? For example, it's a simple permanent tissue. Phloem is a complex permanent tissue. Likewise, they might ask. Okay, it's about different tissues present in a plant. That means with respect to anatomy of a flowering plants. Okay. Then, apart from these tissues, we want to study about tissue systems. Tissue system in anatomy of flowering plants. Tissue systems. Say the first issue we have to discuss about epidermal tissue system. Epidermal. The ground tissue system. The vascular or conducting tissue system. The epidermal tissue system is the top layer, for example, epidermis, the cuticle, the stomata, the guard cells, likewise we have to study. The guard tissue system or fundamental tissue system, we have to study about simple tissues like parenchyma, collenchyma, sclerenchyma. Vascular tissue system, in vascular tissue system or conducting tissue system, we have to study about the complex tissues like xylem, the phloem. So, You have to study and interrelate these different tissue systems for better understanding. This is very important. And another sub-theme in anatomy of flowering plants, we have to discuss about important anatomies. For example, now say dicots and monocots. Dicot stem, dicot root and leaf. Monocot stem, monocot root and leaf. These three, that means dicot stem, TS and its technical description called general anatomy, the different parts. The root, the leaf. Same in case of monocot, stem, root and leaf. These are very, very important. For example, in case of xylem, that means xylem and phloem, vascular tissues. Say they might ask you like this: what kind of arrangement of xylem? That means type of vascular bundle. That means xylem and phloem together considered as a vascular bundle. In case of root, the kind of arrangement of vascular bundle is radial. In case of stem, it is conjoint. Here, stem, here conjoint means if there is a cambium, it is called open because it leads to secondary xylem and phloem. If there is no cambium, it will be closed because there will be no possibility for secondary xylem and phloem. So we have to understand these details. Radial, conjoint, the words, the technical words are very important in understanding these things. That means if we know about technical words, for example, radial vascular bundle will be in root, for example. Whereas in case of stem, it is conjoint. Likewise, we have to interrelate and analyze. Okay, these are about different, the basic keywords in understanding different anatomies. Okay, then it is, as is mentioned, the structural organization in plants may weigh around 9%. Okay, the third unit in +1 is cell structure and function. Cell structure is around nearly 15%. Very important. Cell structure, we have to focus on the basic aspects of cell, like cell theory, for example, Schleiden and Schwann. Cell theory. Rudolf Virchow, he has improvised the cell theory. Say, omnis cellula e cellula. That means the bottom line of the cell theory states that all living organisms composed of cells and cell products, and all cells originated from the pre-existing cells. So at least in the basic level we have to understand what exactly the cell theory states and who proposed it and who improvised this. Likewise we have to understand. Okay, coming back to the other issues, cell, these are two kinds, prokaryotic cell and eukaryotic cell. As I mentioned already, in all prokaryotes comes under Monida, whereas the rest under eukaryotes according to the grade of organization. Okay, we have to understand the basic characteristics of a prokaryotic cell. Say the first feature, a prokaryote, the first nucleus, what we call underdeveloped nucleus. Karyon means nucleus, pro first means it implied that under or poorly developed nucleus. There is no well-defined nucleus. What actually the word, that means the interpretation of the word well-defined nucleus. That means there is no nuclear envelope, there is no double-layered nuclear membrane, and here the nucleus, actually the genetic material is considered as a circular, that means single chromosome or circular chromosome. That means the genomic DNA is nothing but a single chromosome or circular DNA. That's it. And the significance of this prokaryotic cell is outside this genomic DNA or circular DNA, there is a small circular plasmids. Okay? Plasmid. That means the genomic DNA or what we call single chromosome. The entire organization will assume a shape of just a single chromosome. Here the thing is, there are no histone proteins in prokaryotic cell. As I told already, prokaryote first, that means poorly developed nucleus means that means almost all the features are at the primitive level. Advanced, that means histone proteins are essential for DNA packaging. That is why eukaryotes, true nucleus means in eukaryotes there are histone proteins which enables the DNA to pack, and also there is a well developed nuclear envelope and two-layered nuclear membrane. This is the first most distinguishing characteristic feature between that distinguishes prokaryotes from that of eukaryotes. Okay. Then we have plasmids. For understanding, I'll write like this. Okay. The prokaryotes. It is better to analyze the diagrams of prokaryote and eukaryotes for understanding. In eukaryotes, no well-defined nucleus. No well-defined nucleus. It's just in the form of circular DNA or single chromosome. Here, well-defined nucleus and two-layered envelope. Nucleus, that means enclosed by double-layered nuclear membrane. Histone proteins, for example, for packaging. Here, the chief factor is plasmids. Outside the genomic DNA, there is a small circular DNA called plasmid. It gives some important phenotypic features like antibiotic resistance, for instance. Okay, and here, except 70S ribosome, there are no other cell organelles. Here, ribosome is 80S ribosome, and there is a significant cytoplasmic compartmentalization, what we call cytoplasmic compartmentalization. Or balkanization. Cytoplasmic compartmentalization, in a sense, there is a well-defined cell organelle system. That means membrane-bound cell organelles. Cytoskeleton, which gives mechanical strength, the shape. Say cell organelles means, as I mentioned, for example, just discuss. I've just give the names like Golgi apparatus. Golgi apparatus. Packaging system. Packaging what? Such components which are directed for intracellular or for outside the cell. Packaging system, just like postman. Then the mitochondria. Whenever the mitochondria comes, there is a word oxidative phosphorylation. That means synthesis of ATP, the powerhouses of the cell. Ribosome already mentioned on protein synthesis. Then endoplasmic reticulum, it has smooth and rough. Smooth endoplasmic reticulum majorly consists of lipid synthesis, whereas rough endoplasmic reticulum it majorly includes the synthesis of proteins. Lysosomes, that means it consists of hydrolytic enzymes. Peroxisomes, there are several other cell organelles. So here the thing is, in case of eukaryotic cell, in case of plant, there are vacuoles. In case of plants. In animals, there is no cell wall. In case of plants, there is a cellulosic cell wall. This is very important, But just like that, in case of prokaryotes, there is a cell wall, but it is not cellulosic, made up of polysaccharide plus amino acids. And most important, in most of the prokaryotes, particularly the bacteria, they have a complex, that means complex cell envelope. Say outer glycocalyx, cell wall, and plasma membrane. Glycocalyx is based on the chemical composition. It again has slime layer and capsule. Capsule is rigid and tough, okay? Slime layer is somewhat loose. Based on the difference in the composition or the or how the cell envelope responds, bacterial groups can be classified into gram positive and gram negative, whether they absorb the gram stain. So it's about different keywords, but understand at least at that means conceptually at the basic level. The technical terms understanding is very important. They might ask what is slime layer, what is glycocalyx, what is capsule. Likewise, they might ask you, and what is plasmid. Likewise. So technical understanding is very important in these cell structure with respect to basics. Okay. Yes, but the other issues, we have to study about fluid mosaic model. Fluid mosaic model of plasma membrane, at least at the basic level. Then chromosomes. Here chromosomes means, say, in case of eukaryotic chromosomes, I am saying, say, based on the position of centromere or primary construction, they have been classified into metacentric, submetacentric, telocentric, and actocentric. That means actocentric and telocentric. That means based on the position. And kinetochore around the centromere or primary construction. What are centrioles, centrosomes? So the micro bodies you have to understand, and at least at the definition level, so that we can understand what are its significance and functions as well. So it has about different eukaryotic and prokaryotic aspects of cell structure and function. Okay, that means the first basic unit. Apart from the cell structure and function, we have to study about biomolecules. In the last class we have already discussed, biomolecules, the major biomolecules, polysaccharides, the amino acids, the lipids, the nucleic acids. In nucleic acids, say for example nitrogen base, that means purine, that means nucleosides and nucleotide, and that means association of sugar molecules plus nitrogen base nucleosides, for example. And here the nitrogen base again purines, pyrimidines. The purines, adenine, guanine.